This is the fourth out of 4 lectures. I've enjoyed myself immensely visiting this wonderful institution. Joy and I have enjoyed this wonderful city and the hospitality of so many of you, starting with you, Alain, but certainly not limited. And it's been a real privilege on my part to have this opportunity. I also want to thank those of you in the audience. Some of you have been very faithfully coming to several or even all 4 of these lectures, and I can't imagine why you would do that, but I really appreciate that you have. So today is going to be a bit of a departure for me. I usually speak on purely scientific subjects. I won't deviate, as you'll see, entirely from science. I'll talk a little bit about the history of science and medicine, but I'd like to do it from the perspective of illustrating the role that science plays for society and what society expects of science. And the theme that I'll develop is that scientists do science for different purposes, for purposes other than those that society values science for. And so there's an inherent historical conflict, which I think is fundamentally irresolvable, between the people who practice science and their motivations and societies that pay for them to enjoy themselves. And the result of that dichotomy is an interesting dilemma that every society faces, and different societies have had different answers to how they resolve this inherent conflict. And the way in which they resolve this conflict, I would suggest, is a major determinant, possibly the major determinant, of their prosperity long-term as a society. I'll also develop the theme that societies can change the way that they balance this dialectic, in a way, between the impatience of society and the patience of a successful scientist. And so you can have, and there are examples today of societies, unfortunately, that are shifting in the way they value science, and I believe will pay correspondingly a price in the long run in terms of quality of life. So those are the themes that I'd like to illustrate today. And I'll begin with some thoughts on the nature of a scientific discovery as distinct from the nature and the process of the discovery. So a discovery in science, what is the result of that discovery? I think we might all agree that it helps us rationally organize information, and it increases our ability to predict outcomes in the real world. That's essentially what science does, and it's for that reason that science has been such a successful strategy for harnessing the human intellect. If you think back over millennia, at first we had religion, then we had philosophy, and when both of those approaches failed to produce results in the real world, we discovered the method of science. And as Steven Weinberg wrote in a very impressive book that I had a chance to read this summer, the discovery of the method of science is actually the greatest discovery ever in science. So, armed with the ability to organize and predict information, science has taken off as an enterprise, and society has valued that enterprise because of the technologies that have resulted and the improvements in the quality of life that have resulted. And yet, the discovery of science is carried out by human beings. The discovery of science is therefore a human process, and because it is carried out by us, it is inherently unpredictable, because we are unpredictable. We like to think that we are rational, et cetera, but in fact, the process of science, as history shows over and over again, is impacted by personality, the individual, circumstances in which the individual finds himself or herself, which I'll refer to as scientific culture, that relates to the the value that science in your society or mine or anyone's society, they differ, place on the scientific enterprise, and also relates to who our teachers are and who their teachers were. And it's therefore a multi-generational set of circumstances. And of course, the emotions that we bring to the process of discovery, which is, of course, as every scientist knows, fraught with difficulties, driven by intuition and also ambition. And then the other theme I'd like to develop with some examples is that the long-term impact of a discovery is equally unpredictable. It has been almost impossible to gauge the impact of important discoveries. If a discovery or a finding is relatively modest, well, sure, you can predict what its outcome is going to be. But if it's a major discovery that changes the way people think, oftentimes it's not fully appreciated for many years, even decades, even today. And so the impact of these discoveries becomes inherently unpredictable, which means that any strategies for funding science need to take into account the fact that intended consequences are rarely the actual consequences. So let me illustrate this, these concepts, and then at the end, at the end, I'll turn to the implications for how societies can productively harness science for their benefit. But I'd like to illustrate this with a very tangible example, which is the basic science that resulted in our ability to control cholesterol today. So there's what I'll spend the next probably half an hour with you doing is reviewing the history which required 100 years of discovery that has resulted in something we take for granted today. I suspect that a fair number of you, at least those of you over 50, are taking a drug called a statin to control cholesterol in your blood. And the reason I take it, and the reason that we take this drug is that it lowers lipoprotein levels in the blood, and this has been proven to extend lifespan by reducing heart attacks. Now, actually, this 100-year journey begins right in this very city. In 1897, not far from where we stand, in the work of the Curies and the Becquerel. And I'll describe that momentarily, the impact, unexpected impact. So let's take a look at the situation with respect to cardiovascular heart disease, even 50 years ago. In the 1960s, it was very common for relatively young people, especially men, to die of heart attacks in their 50s and 60s. I grew up in the 1950s and 60s, and many of my friends' fathers died in their 50s and 60s, otherwise healthy people, of sudden heart attacks. This was really very common. In fact, the death rate from coronary heart disease rose steadily throughout the 20th century, and it peaked in the late 1960s. Now, today, the toll is much less. There, as you can read here, there are about 400,000 deaths each year. I'm referring now to the United States, versus an expected 1.7 million that would have occurred had we followed the same curve through 1968. So something happened that drastically, by a factor of about 3 to 4, reduced early deaths due to heart attacks, and also strokes, reduced 75%. And altogether, this adds an average of 5 years of life expectancy to both men and women. So these are extraordinary changes in demography. How did it happen? Well, there are two developments that came from basic science and which translated into drugs. Drugs that control cholesterol and drugs that control blood pressure. Together, these two, the ability to control these two conspiratorial pathophysiological axes that conspire together to weaken our vascular system have resulted in these enormous changes in public health that we all benefit from today. I won't have time to talk about the history of how drugs that control blood pressure came about through the discovery of the physiological mediators of blood pressure, the receptors, that control them, and ultimately drugs that target them, recognized very recently in 2012 by the Nobel Prize in Chemistry for the discovery of the G-protein coupled receptors, which are the targets of these drugs. But I won't have time to go through that story. But if I did, it would sound an awful like the story that I will tell you of how we learned to control the body's level of cholesterol, and therefore to control, to a large degree, heart attacks. Now, this story is a story of basic science that, as I will illustrate, emerged over more than a century in almost entirely unpredictable ways. If you had started out, let's start with the man who actually discovered the drug. His name is Akira Endo. He was working in Japan in the 1980s, late 1970s and early 1980s. And he was able to sift through 6,000 or 7,000 strains of microbes to find the rare fungus that secreted a product that could inhibit an enzyme that was important in the synthesis of cholesterol in the body. And he was able to isolate that compound, determine its structure, and ultimately test it in animals and lead the development in humans to show that it lowers blood cholesterol. Now, for that, he was recognized by the Lasker Award, the major American prize in medicine in 2008. And his work, as I've indicated, began in the 1970s. And the enzyme that is the target of the inhibitor he discovered, so this is the fungal metabolite that Endo discovered. He named it compactin. It targets a specific enzyme called Hmg-Coa Reductase, hydroxymethylgluteryl-CoA reductase. I'll have more to say about this enzyme later. And it prevents its conversion to a later intermediate, methylonic acid, which ultimately results in the synthesis of a very complex molecule called cholesterol. Now, the ability of this drug, of Endo, to discover this drug, of course, required a lot of prior knowledge. He had to know that if he were to inhibit this enzyme, it would really make a difference in the body. In other words, that it is a valid target for pharmaceutical intervention. So this enzyme had to be discovered, which meant this pathway had to have been discovered, which also meant that the structures of these molecules had to have been discovered, including the structure of cholesterol itself. And it also meant that basic tools in the life sciences that allowed, in the 20th century, the glory era of biosynthesis, the ability to discover the basic principles by which energy from food is converted into carbon skeletons to make up our body's material. All of that actually had to happen first. At the apex of all of that came his important discovery of this inhibitor, which was later developed within a few years by Merck into the first statin, and then later further modified by many other pharmaceutical industries to all of our benefits. So what then enabled Endo to discover the statin drugs in the early 1980s? I can bring you back to Paris then in 1903, the Nobel Prize in Physics, which recognized the discoveries only a few years before of these very familiar figures, who discovered radioactivity and learned how to isolate radioactive material. Now, the existence of radiation, of course, was entirely unexpected. And who would have thought that then, at the turn of the 20th century, that the discovery of radioactivity in Paris would lead within a few decades to the unraveling of the key metabolic pathways that are responsible for life in every living organism on the planet. A key step was taken, as recognized by the Nobel Prize in Chemistry in 1943, by de Hevesy, who was the first to recognize that isolated radioisotopes could be used in the body, or in his case, in plants, to trace the movements of fluids and materials within the body and establish precursor product relationships. The long-term impact, though, of this discovery was in the hands of the biochemists. And you'll see a little bit later in the 1940s, in the early 1950s, that biochemists later in New York were in fact able to take radioisotopes and use them in entirely innovative ways to trace the synthesis of the backbone of molecules like cholesterol. And as a result, they and others taught us the basis of, to understand the basis of life in chemistry. I'm quite sure that no one in 1900 could have predicted that essentially the development of our understanding of biochemistry and molecular biology and ultimately genetics was based on the discovery by Becquerel and the Curies in Paris. But this illustrates that the most fundamental discoveries have the most unpredictable and the most important impacts. Well, let's return now to the story of cholesterol. Cholesterol was actually discovered by Wieland and Windaus, working independently in Germany, in Munich actually, Munich and Göttingen. And they were recognized in 1927 and 1928 by a joint Nobel Prize. Many of us who've received Nobel Prizes have wondered how the Nobel Committee comes to its conclusions. Apparently in 1927, for some reason, they were unable to come to a conclusion, and they reserved the Nobel Prize, and they managed then to combine two Nobel Prizes for the same topic. Somehow the logic of this is lost in history. But what they did recognize turned out to be very important. Wieland and Windaus were were natural product chemists. There was a school of natural product chemistry that dates back to the mid-late 19th century in Germany. It gave rise to and was motivated by the dye industry, for example, in Dresden, where dyes could be, there was a drive to synthesize dyes and make new colors, a purely commercial interest, but actually it gave rise to what we now recognize as the field of organic chemistry. And it gave rise, as a result, to the first demonstration and understanding of chemical structures. Out of that culture arose the first biochemists, which is why Germany was so powerful in biochemistry. It had developed a scientific culture already at the turn of the 20th century that was several decades old, that was driven by structure, that drove the understanding of structure and reactivity, even before we understood the nature of a covalent bond in terms of orbitals and the fundamental rules. Well, this culture of natural products turned to medicine. And of course, we also make natural products, and among these are the bile acids, and among these are cholesterol. And the pathologists, even then, had been looking at atherosclerotic lesions from patients that had had heart attacks and discovered that they contained compounds in them that were, roughly speaking, similar to the compounds in the bile and to other compounds in nature. And so the great organic chemists / biochemists of the time turned their attention to this class of compounds and recognized that they had fundamental relationships to each other. And that made them interesting because it meant that there was some universal feature of these compounds in all of life. And out of this curiosity came, in the hands of these individuals, the discovery of cholesterol. They discovered essentially the carbon skeleton of cholesterol, and they recognized, obviously, it was an enormously complex structure. At the time, it was impossible for anyone to imagine how an organism could synthesize this molecule or anything nearly as complicated. You have to remember in 1920 or 1925, actually we didn't have any idea of how a sugar was broken down, never mind how it was made, and that's only 6 carbons. This seemed, and it led actually to a dogma, and the dogma was that essentially you are what you eat. There is simply no way that your body could make cholesterol. It had to come from somewhere, okay? Which meant that if that were correct, no drug could influence how much cholesterol we have. It's only what we eat. Well, of course, that turns out to be profoundly wrong. But what I'd like to do, rather than talk about the science they did so many years ago, is ask what motivated Windhaus and Wieland. And I must say, I found the Nobel lectures to be a very useful historical resource. You can kind of go back contemporaneously. In those days, Nobel laureates were often recognized 2 or 3 or 5 years after a discovery was made, rather than 15 or 25 or 30. And so they actually remembered what it was that they did, okay? And they remembered what motivated them at the time, unlike the situation presently. And so here's a quote from Heinrich Wieland. "The task would appear to be a long and unspeakably wearisome trek through an arid desert of structure," written as only a German of his time could write. "But the driving force which steels the perseverance," you get a feeling that it's tough for Heinrich to show up in the lab, "lies in the problem itself." Now, I happen to know a little of Heinrich Wieland because he is the grandfather of one of my closest friends, Felix Wieland. And I do know that Heinrich apparently had very little sense of humor. And that actually shows here. But what does show is his motivation of the problem in the problem itself. He was actually apparently such a modest man that he snuck off to Stockholm and did not tell his family that he was receiving the Nobel Prize. His children grew up actually not knowing that their father was a Nobel laureate and found out about it only in school and when they discovered that there were streets named after him. Well, anyway, the situation seems to be a little different today. But the important point here is what motivated him was the problem itself, the challenge, okay? That's what motivated this scientist. So, now we know what cholesterol consists of. Where does it come from? Well, to leap ahead a little bit, it comes from 5 carbon units. There is a basic building block in life called isoprene, 1, 2, 3, 4, 5 carbons. That 5 carbon unit can be put together into polymers of the 5 carbon units by joining carbon to carbon. So this is 2 isoprene units put together to make a 10 carbon unit. And this can happen over and over again. Out of 5 carbon units ultimately come cholesterol, bile acids, which are derived metabolically, as we now know, from cholesterol, and as well many other important compounds, including rubber, vitamin D, and not least of all, in case you need this to wake you up, the sex hormones, testosterone and estrogen. And in fact, it was another German biochemist and a colleague, Butenandt and Ruzicka, who discovered in the 1930s the sex hormones, testosterone and estrogen, and in the process discovered their common biological origin in relation to cholesterol and the rubber terpenes, all from the same 5 carbon building block. So this important discovery not only gave us an understanding of sexual differentiation and important physiology, it also gave us the first steroid hormone drugs, leading to later steroid hormone drugs that we use as anti-inflammatories. And it also gave us the basic understanding that all of these compounds are derived from a common precursor consisting of 5 carbons and isoprene unit. So this was the next important landmark. Well, now we have to get a little more fundamental because we have in our head the idea that there are 5 carbon pieces of which quite a number of them will be added together to form this much larger cholesterol molecule, but we don't know how. And actually, we are missing a even more basic concept. How does the body synthesize carbon skeletons at all? If we don't understand that in general, we surely could not understand how a very complex carbon skeleton is made. And of course, you might think, and it's true, that it takes energy to bring together 2 carbon atoms to make a carbon-to-carbon bond. And so there will have to be an investment of metabolic energy to do that. And the key to understanding how all of this works is to ask a more basic question. And something that you learn over and over again in the history of science is that even the most important problems that have the most practical concerns will come to an absolute standstill if a basic concept is missing that needs to be solved in order to remove a roadblock. Now, the problem is that most of the time when that happens, we don't know what that roadblock is, actually. That's one of the reasons why science is so unpredictable. We don't even know how to formulate the question. It's only the greatest of scientists who are able to formulate the question in a way that allows it to be recognized as a key question. Well, in this case, the question is, how is a hydrocarbon unit of any kind, activated energetically to be able to be grafted onto another one to make a more complex molecule? It is actually the most fundamental question in all of biochemistry and probably one of the most fundamental questions in all of life. This discovery was made by perhaps the greatest biochemist of the 20th century, Fritz Lippmann. And like Albert Einstein, probably the greatest physicist of the 20th century, his Nobel Prize recognized a very limiting feature of his contribution to science. What Lippmann actually conceptualized in the 1930s was the idea that metabolism, that when we oxidize sugars, the very purpose is to capture energy in an intermediary molecule that is then saved and used to invest that energy in the synthesis of skeletons like carbon-to-carbon bonds. And he deduced that that molecule is ATP, as the principal energy currency of the cell. Those of you who have studied biochemistry know there's a concept called high-energy phosphate, and high-energy bonds, low-energy bonds. This was Fritz Lippmann. The high-energy form of phosphate, squiggle P, that was a notation that he introduced. And out of this came the whole fundamental connection of thermodynamics into biochemistry and into life. And so our whole view of what a cell does and what a body does was altered with this concept. Now, out of that view came experiments that he carried out in the simplest possible way to ask, how can carbon be activated to make a more complex molecule? And in the process, he discovered the key molecule for activating carbon in the cell called coenzyme A. And you'll see that I'm not really taking a detour because coenzyme A, you'll see in a moment, is used over and over and over again at every step in the synthesis of cholesterol, whose pathway could not have been discovered, nor could the drug have been discovered without this foundational concept. So what is it that Lippmann did? And what he did here really illustrates the style of biochemical discovery in the middle of the century, the 20th century. So I'll go through it here. He had the idea that there had to be a way to activate a 2-carbon unit. Now, everybody here knows what vinegar is, but those of you who've forgotten your chemistry probably don't remember it's acetic acid. Acetic acid has 2 carbons. So when acetic acid is added to another molecule to acetylate it, this requires taking organic 2-carbon units and forming a carbon-to-carbon bond in an acceptor molecule. And what Lippmann did was short-circuit the entire process of metabolism, because very little about metabolism was known, and he took acetic acid in a test tube. He added a soluble extract of liver to it, just liver juice. He added ATP because of his incredible insight that ATP provides the source of energy. And then he had to add something for the acetate to go onto. So he added a sulfur-containing compound that was known by doctors to be metabolized in the body by having 2 carbons added to it. And it was known that it was metabolized in the liver, but it was of no importance to the normal metabolism that goes on in the body. But he had the insight to add that to this incubation. And what he found was that acetate groups were readily transferred to this drug in vitro, and it required ATP. And it required 1 molecule of ATP for every 1 molecule of acetate that was added. He found that if the extract was dialyzed, it lost its ability to be acetylated. And then he boiled the extract, which is a trick introduced many years earlier by the great biochemist Hardin, who discovered coenzymes. And he found that the boiled extract, though it was inactive by itself, could be added back to the dialyzed extract and this would reproduce and reconstitute acetylation activity. So the boiled extract must have contained a heat-stable, which means non-protein, small molecule that conferred the ability to transfer acetate in this ATP-dependent way. What was that molecule? Well, over the next several years, he and his colleagues, Lippmann and his colleagues, isolated and purified this from these boiled extracts, and were able, through arduous process, to purify it about 1,000-fold and establish the basics of its chemical structure. He called it coenzyme A. It was a coenzyme in the sense of Harden and Warburg, and A for acetate, because he suspected that it was a general cofactor for the activation of carbon in the form of acetate. This is its structure, and as you'll see, this sulfur actually forms a bond to the carboxyl group of the 2-carbon acid, acetate, and this is indeed a high-energy form of a high-energy bond. That's how 2 carbons are activated. Now, the main point here is not to explain the history of the science, except to say why this was important. But what enabled Lippmann to do this? Why? Well, I mean, he was a genius. That, of course, is a predisposed condition that made it possible for him to do it. But you know, the world is full of geniuses. There are probably 10 of them in Paris today, okay, or whatever, and probably an equal number in Bangladesh. But I'll bet you the ones in Paris are more likely, if they stay in Paris, to discover something than the ones in Bangladesh, unless they come here. And so what was it about Lippmann that enabled him to do this? Circumstances within a scientific culture. This is a quote from his Nobel lecture. After my apprenticeship with Otto Meyerhoff, Meyerhoff discovered the so-called Embden-Meyerhoff pathway, which we now call glycolysis. So he, in the 1920s, made very important discoveries on the metabolism or catabolism of sugar in muscle. After his apprenticeship with Meyerhoff, his first interest became the Pasteur effect, this peculiar depression of the wasteful fermentation in the respiring cell, by looking for a chemical explanation of the mechanism of pyruvic acid oxidation. So he picked up his interest from the scientific culture at the time. It was, of course, a German culture, and also in earlier times, a French culture. Also, accident. You will all remember the very famous quote of "chance favors the prepared mind," which very much applied to Fritz Lippmann. This decision started me on a rather continuous journey into virgin territory to meet with some unexpected discoveries, the most important being the accidental observation that oxidation was completely dependent on inorganic phosphate. Okay? That was a key discovery that led to this breakthrough in bioenergetics and in understanding how carbon skeletons are formed, and which truly opened the glory years in which virtually all of the cell's constituents, whether they be proteins, amino acids, nucleic acids, DNA, RNA, sugars, carbohydrates, fats, membranes, the synthesis of all of these compounds was to become elucidated in the following 15 years as a result of this epochal discovery. So how then does coenzyme A work to activate acetate, and how does this result in cholesterol? Well, we are now turning to the Nobel Prize in Physiology or Medicine in 1964, which recognized the work in the previous 10 years of a German-born biochemist, emigrated to the United States. I'll describe that in a moment. And Fyodor Lunin, Konrad Bloch and Fyodor Lunin, who worked, in fact, who inherited the labs of Meyerhoff and Warburg and Wieland, and was a student of Wieland. So this work took place in Germany. This work, in a way, began in Germany because Bloch was a student of Wieland. So again, it's the culture, but in this case, the culture has begun to migrate. And they were recognized for their discoveries concerning the mechanism and regulation of cholesterol and fatty acid metabolism. And essentially what they discovered was how the coenzyme A was utilized to lead to the synthesis of cholesterol. It was actually Lunen competing with Lippmann who discovered that acetate is incorporated into coenzyme A as part of the biosynthetic pathways that Lunen and Lippmann went on to discover more of which I've mentioned a moment ago. So Lunen had an important contribution in understanding and discovering the direct linkage of acetate to coenzyme A, and indeed made major contributions to the discovery of the coenzyme that Lippmann discovered, to the elucidation of the structure. Now, what Lunen and Bloch did, working independently, but with a lot of back and forth in terms of contemporaneous discovery, is they discovered how the 2-carbon unit of acetate is made into the 5-carbon unit. You remember, it's been a long lecture, but you remember a few minutes ago, I said that isoprene was very important, okay? And Butenandt had discovered that the 5-carbon unit isoprene was the common precursor of Windaus and Wieland's cholesterol molecule. So that left us with the question, specific question, where does that 5-carbon unit come from, and how are those 5-carbon units put together to make cholesterol? And what I said was we needed to do a detour to understand how any kind of carbon comes together, and now we've made that detour, and now we're back to the main track. So how then is 2-carbon unit of acetate made into the 5-carbon unit isoprene, and how are N of those put together to make cholesterol and the steroids, where N is equal to 6, and rubbers, where they're much longer polymers, polyisoprenes, which are used in many other aspects of biosynthesis, including, by the way, mitochondrial bioenergetics, where they act as a coenzyme in carrying electrons, and they're used as carriers in cell wall synthesis, in bacteria, for example, and vitamins, like vitamin D. All of these compounds follow this pathway, but they branch at certain stages. So how then does this occur? Let's start with, how is the 5-carbon unit made from acetate, the 2-carbon unit? It's a complex pathway. But if you forget the carbon skeletons for the moment and just focus up here, the overall idea isn't complicated. You take 3 2-carbon units, which of course makes a 6-carbon unit, and you knock off one of those carbons as carbon dioxide, thus leaving a 5-carbon unit. That is where the 5-carbon unit comes from. And if we follow this pathway around, we start with acetyl coenzyme A. We put 2 acetyl coenzymes A's together. That makes 4 carbons. We add in 2 more carbon units from acetyl-CoA, the energy in each case coming from hydrolyzing the linkage to coenzyme A. That high energy bond provides the energy to graft that carbon onto the growing skeleton. This makes a 6-carbon unit. You don't have to be a chemist, and I'm not, to understand 2 plus 2 plus 2 equals 6, okay? Now, you do a little gyration that I won't go into, and you knock off a carbon to make a now high-energy intermediate called isopentyl pyrophosphate. Those 2 phosphates each come from ATP and are later removed as a pyrophosphate unit in order to graft one 5-carbon unit onto another, but I won't show you that. But it does explain why you need ATP in this pathway, and how it is that inorganic phosphate is incorporated into the carbon skeleton as a way of retaining the high energy state in this 5-carbon molecule. Now, the only reason to show you this slide, which looks very complex, is that embedded within it for the first time is the enzyme Hmg-Coa Reductase. So it is only in the late 1950s, early 1960s, that for the first time humans understood that there was an enzyme called Hmg-Coa Reductase. And it is the first time in history that we understood that this enzyme is important in the synthesis of cholesterol. But there was nothing at the time to distinguish that enzyme from any of these other steps or any later steps in the synthesis of cholesterol to know which, if any of them, might be worth interfering with in the body to control the level of cholesterol. Okay? That was not known. Nor was it known that controlling the level of cholesterol would actually make a difference in terms of health. Okay? So I've taken you all the way from 1897 to about 1960-ish, right? That's a long ways, 60 years. And it's only now that we know this enzyme exists, but we still have no idea. It has not risen out of the woodwork as being of any medical importance whatsoever. What motivated Bloch and Luhnen? Okay, and I'll come to the rest of the story in a moment, but I want to deviate because an important idea I want to get across is what motivates the scientists. Okay, once again, the problem itself, as Wieland said. This is what Conrad Bloch wrote. To chemists and biochemists alike, the structure of cholesterol at once presented a biosynthetic problem of exceptional challenge. He did this because it was an exceptional challenge. It was a challenge to him personally, it was a challenge to the field, and it was a matter of a personal ambition to conquer a mountain. Okay? That is what motivated him. Fyodor Luhnen. Once again, he was motivated by the circumstances within a scientific culture. My subsequent concern with the problem of acetic acid metabolism arose from my studies in Heinrich Wieland's laboratory, a laboratory that he ultimately took over when he became Geheimrat. So, that's what motivated these people. Now, a little bit more on the scientific culture that enabled both of these great scientists to do their work ultimately on two different sides of an ocean. Start with Luhnen. Luhnen trained with Wieland and he stayed in Munich. Bloch was a student of Wieland but was unable to complete his graduate studies in Munich because he was Jewish and he needed to flee for obvious reasons in the run-up to World War II. He was successful, actually, in doing so because Geheimrat Wieland helped him get out. And I know this from reading Conrad's biography. Helped him escape, actually, to Basel, where he continued his studies briefly. And he arrived at Columbia University in New York. He actually was given a visa. It's an interesting story. He was given a visa because he had a job offer at Yale, where I'm now a professor, okay? And Yale was never known for its generosity. In fact, the first Yale professor to ever receive any salary at all was the very famous physicist J. Willard Gibbs. He came from a poor family. He actually needed to earn a salary, and he demanded one. Happily, I can say we all are now paid. But he was the first actually American academic who received any kind of a salary at an Ivy League school, at least. Well, Conrad received an offer from Yale to join the Department of Biochemistry or biology, or whatever it was called at the time, in the School of Medicine. He received that letter from the department chair. The next day, he received a letter from the dean of the school, which said, oh, by the way, this professorship comes with no salary. So young Bloch apparently had the good judgment to show to the immigration officer the one letter but not the other. And so he was granted a visa to come for the job. He also had the good judgment not to go to Yale. So he went instead to Columbia, where he had in hand, I mean, you can't make this stuff up. He had in hand a letter from Heinrich Wieland, who, as I've already told you, was not a man of great humor and also of many words, as I described, okay? And the letter, when it was opened by Professor Schönheimer, another German emigre who came from Berlin, also Jewish, to New York a few years earlier and had established himself at Columbia, Schönheimer opened up the letter, which said only 4 words, which is apparently all you needed to say in those days, especially if your name was Wieland. Herr Bloch ist gut. That's all it took, 4 words. So Herr Bloch was good. And he also had some good luck, because Schönheimer was connected at Columbia University to the great chemist Harold Urey. And now Urey discovered heavy water, D2O. and he learned how to isolate it, actually in 1932, recognized by a Nobel Prize in 1934. That's how quick things seem to have been in those days. And Urey was put together, actually, Schonheimer was put together with Hevesy, the person I mentioned earlier, who had the basic idea of using tracers, and they had met in Germany. So when he came to Columbia, and he became aware of heavy hydrogen and the fact that it could be purified, he got the idea, the really epical idea, that tracers, in this case stable isotopes, could be used to trace metabolic pathways. One of his first students was Conrad Bloch. Now, unfortunately, Schonheimer died very young. He committed suicide, although clearly he was a great biochemist and maybe had one of the most important ideas in the history of biochemistry. He was unfortunately a chronic depressive, and he committed suicide in his early 40s. But this left a laboratory which was taken over by Bloch at first, and Bloch then was in a position to use stable isotope labeling to develop a detailed understanding of the synthesis of cholesterol. And I should say that Lunan also picked up on this technique, but in his case used radiocarbon, which became available after World War II in the development of nuclear reactors. So you can see that all of this ferment occurred in a historically rich context that had great consequences for the scientific cultures, both in Germany and in Europe and in the United States. So how then, coming back to where we are in the story, we now know how cholesterol is synthesized, but we actually don't know how the body controls its cholesterol. We know the body can make cholesterol. We know the pathway by which cholesterol is made, but we don't yet know where in this Bloch-Lunan pathway could be a target for controlling cholesterol with a drug. And we also don't know whether excess cholesterol actually causes heart disease. There was, at that time, even as recently as the 1980s, a large accumulation over more than 100 years of evidence that was correlative. Lesions that caused heart blockages have a lot of cholesterol and cholesterol esters in them. People who had higher fat diets were more prone to heart attacks. But there was very little actual proof, in fact, no proof, that controlling cholesterol would actually control heart disease. The actual proof has only come from statin drugs, where large populations have been in controlled studies and in population-based studies been on statin drugs, and their cholesterol is controlled, and the rate of heart attacks is greatly diminished. So we know this for a fact today. But really the first strong proof, genetic proof, came from a disease called familial hypercholesterolemia. This is a disease that is fortunately rare. It's a homozygous, well, it's called a heterozygote disease because if you have one copy of the bad gene, you're going to have a high blood cholesterol problem your whole life, and without pharmacological therapy, which happily we now have, in the natural history of the disease, you would probably die in your 30s or 40s of a heart attack. If you have a homozygote form of this disease, you will develop lesions like this and likely die of a heart attack before you're 15 years old. So it's a very, very striking disease. The gene was identified in its molecular nature was discovered by Michael Brown and Joseph Goldstein. And in the process of this discovery, they also discovered how the body regulates cholesterol. And so at one time, with one major discovery, they proved beyond a doubt that if you could control the level of LDL cholesterol in the blood, you would be able to control the rate of the incidence of heart attacks in the population. And they also discovered the means to do this by controlling Hmg-Coa Reductase. Through studies of fibroblasts from diseased patients, they discovered that the cholesterol in its main form in the blood, called low-density lipoprotein, in which cholesterol esters are carried inside of a lipoprotein particle, they discovered that these are metabolized by cells in the body by first being taken up into the cell into lysosomes, the known degradative organelle. And this uptake is highly selective and mediated by a receptor they discovered which they called the LDL, or low-density lipoprotein receptor, that takes up the lipoprotein particles in vesicles, delivers these by membrane fusion, the very process that I talked about in my first lecture, for degradation and release of the cholesterol. When that cholesterol is released, it is reutilized at the cell membrane where most of the cholesterol is, and if there's an adequate supply of cholesterol, the cholesterol, they discovered, actually is delivered in a different form, in a metabolized form, to the endoplasmic reticulum, where Bloch's enzyme pathway is localized, and this leads to the repression of the enzyme Hmg-Coa Reductase. It also leads to an increase in the synthesis and expression of LDL receptors. So to summarize, when you have a lot of cholesterol in the blood because you just ate a meal that's rich in cholesterol, what happens is you preferentially will take cholesterol out of the blood and utilize it for the cell and the body's needs, and you will slow down the synthesis in the body of your own cholesterol. After all, why make it if you're eating a lot of it? But on the other hand, if you're not getting enough cholesterol in the body, something that in my judgment would not happen in this country, what happens perhaps to other people is that you don't have enough of these LDL particles. Now you actually de-repress and make more of this enzyme Hmg-Coa reductase, which ratchets up the speed of cholesterol synthesis and allows you, mostly in the liver, to produce more LDL. Okay, I hope that's clear. Now, an interesting feature of this pathway is that when you have a lot of cholesterol, you also make more LDL receptor gene product, and therefore more LDL receptors, which gives you the means to take up even more of the LDL in the blood. So this is how LDL in the blood is measured. So from this basic physiology came the understanding that Hmg-Coa reductase is a key target because, after all, the body regulates the level of that enzyme when you have too much or too little cholesterol. They furthermore discovered that if you artificially lower the level of Hmg-Coa reductase, for example, they discovered this can be done in several different ways. When that happens, you make more LDL receptors, as I've said. When you think about the pharmacological consequences of that, it means the following. If you had a drug that took out Hmg-Coa reductase, knocked it down, you would also induce, because you wouldn't have enough cholesterol, even though the body was trying to make more of it, you may be making more enzyme, but every copy of the enzyme you're making is inhibited by a drug. So as long as you have enough drug, you will not make cholesterol, but you'll continue to make LDL receptors, which means that the LDL receptors will actually help lower the cholesterol because you'll make more than normal of them, amounts of them when you have the drug, and therefore remove more than normal amounts of LDL from the blood. So there's a double whammy that comes about from having a drug that controls cholesterol. So this is the discovery that Brown and Goldstein made. All of which together, finally, in the mid-1980s, positioned endo to be motivated to screen fungi for drugs that would block Hmg-Coa reductase. It enabled him to isolate the enzyme Hmg-Coa reductase, to place that enzyme that's purified in the test tubes, and to add extracts of fungi. Interestingly enough, it was a penicillium organism that gave rise to this drug, a very close relative to the fungus that Fleming first isolated penicillin from many years later. So, reviewing then the history of this discovery, it began with the discovery of radioisotopes in the late 1800s, which no one would have thought would allow the unraveling of the mechanism of the basic discovery of energy conservation and the synthesis of carbon skeleton compounds in living cells. And next was the discovery of the chemical structure of cholesterol. Then the common origin, a broader view of cholesterol and steroids, and most importantly that they come from 5 carbon units. The general principle of energy utilization in biology, and more particularly the way in which this is harnessed to produce hydrocarbon skeletons. Specifically, how coenzyme A activation of 2 carbon units is used to produce the 5 carbon unit, and the key role of the enzyme Hmg-Coa Reductase. The basic mechanism of control of cholesterol by the body, pointing to Hmg-Coa Reductase as the correct place to intervene. And then finally, and only then, was it possible for a drug to be discovered in a targeted way to lead to the improvements in human health that we all appreciate. And so the lesson is that the first nearly 100 years was devoted to untargeted research, made oftentimes with results often by accident, in unanticipated ways, which only then positioned targeted translational research for any hope of success. If Endo had tried many, many years before to have, for example, in the era of penicillin, had he taken the extracts of 10,000 fungi and injected them into rabbits and asked, does cholesterol get lowered? Well, actually, he probably wouldn't have known to look for cholesterol. He wouldn't have known that it would have made any difference if he had. And he probably would have poisoned the rabbits with his fungal extracts because he wouldn't have known how to partially purify them according to some idea. There was no idea. So what lessons can we learn in conclusion from this case study? How can we generalize from it? Well, I've emphasized several times already the importance of scientific culture and apprenticeship. So I want to go back and revisit the names that I've mentioned up till now. And what you'll see is they are all descended from actually a single scientist, von Baer, one of the descendants of the von Baer, of the German dye chemist, one of the great organic chemists in the history of the 19th century. Wieland, Lunen, I think I've lost Bloch in here, but I think, where did Bloch figure in? Bloch worked with Wieland, at least briefly. This is a table put together by Hans Krebs, who, as you'll see also of the Krebs cycle, derives from the same lineage, also a family that left Germany for England, in his case. He wrote an article, a very interesting article, on the making of a scientist, in which he emphasized, when he wrote it in 1967, the importance of scientific lineages. I've actually extended this since 1967. Stadtmann trained with Lippmann, and Brown and Goldstein trained with Stadtmann at NIH. And I might mention, since I'm here giving the lecture, though I didn't make any contributions to cholesterol, although I've certainly utilized my share of it, I worked actually with 2 branches of this lineage, and so this lineage continues in many other fields. And what's important here is not this particular lineage, but the appreciation that if you take any major discovery that was made in any field of science, you will find that there's a lineage very much like this, and you don't have to focus on Nobel Prizes. It's a convenient way to do it, but there are but whatever your descriptor or is of the importance of a discovery, it doesn't matter what it is. It will always end up being a scientific culture, and that scientific culture will always be multi-generational. It does not come out of the woodwork overnight. It is not created when suddenly someone decides to fund science for 3 or 5 years. Okay, it can go away if someone decides to unfund science in 3 or 5 years. That can happen, but it is not created. It really probably takes 20, 30, maybe 50 years to establish a potent scientific culture. So let's come back to the first idea that I introduced, and let's see what, in light of the case study that I offered you, what some of the conclusions we can come to about science and society might be. A discovery in science increases the predictable and increases and rationally organizes information. A discovery of science, as I think I've amply illustrated, is a human process that is unpredictable because it is impacted by personalities, circumstances, and emotions. The long-term impact of a discovery is also unpredictable. So what does this mean? So let's first take the point of view of what motivates societies to pay for the type of science that patiently resulted in these life-changing discoveries over the course of 100 years? Well, we feel, all of us feel, as members of society, an urgent need. We feel an urgent need for specific technologies. I'm trying to broaden this outside of the life sciences. Specific technologies and/or medical treatments which yield improvements in life. And when we invest in these, we want them to have predictable results, and we want them sooner rather than later. I think everybody can agree on that. I think that's why societies fund science. I'd like to think that it's because of the value and the beauty and the scientific culture, which we all appreciate. But I don't think any of us are naive enough to think that that is the principal purpose that taxpayers pay for science. What motivates scientists to do science? Well, over and over again, we've seen the challenge of the problem itself, the basic understanding it creates, the beauty that it reveals, the personal ambition that can be expressed in one way or another. And this is independent of how much time is required. Yes, we'd like to get it done sooner rather than later. We all have a sense of urgency. But sometimes we recognize that important things happen in their own time. And we try to minimize that amount of time, but we have to be patient. And the art of being patient is actually part of being successful in science. So we have an inherent contradiction then between the process of science and society's expectations of it. Science relies on individual taste and initiative. Discoveries are often made by accident. Science relies on unrelated discoveries from other fields in unanticipated ways, which makes the important discoveries in science unpredictable results obtained with unpredictable speed, having unpredictable consequences. It's exactly the opposite of what science wants, and yet that is the only thing that science has ever been able to deliver. So, what are the implications for the funding of research? Science relies on the individual. What that means is it's a bottoms-up process. It's individually initiated. Top-down goals and forced collaborations will never work. Well, they may work to generate resources from the government, but his history shows over and over again that they are doomed to failure in terms of progressing science overall, not in narrow, small lacunae in which most of us operate on most days, but in ways that really matter in the long run. Science relies on chance and unrelated discoveries. Targeted research, questionable. If you wanted to target cholesterol, if you even knew about it in 1920 or 1930 or 1950 or 1970, or even in 1980, it couldn't have been done. You could have had a moonshot project organized around it. It would have been a waste of money. But when the time was right, it happened in 2 or 3 years. And nobody had to target it because it became of interest to the companies themselves. There was a profit motive. The government didn't have to pay for that when it was ready. Science relies on deep multi-generational culture and apprenticeships, which implies that despite all of the reasons why society doesn't like to fund science in the way that it is required, it needs to, and it needs to be steady at the wheel over many decades. So this brings me then to patience and urgency, which is my way of describing the balance between, both at the level of the individual science, but also at the level of a society, between the patience that is needed for good science in the long run against the urgency of short-term needs. And it is that balance that may be the major factor that determines a society's capacity to innovate and therefore to compete. And so with that in mind, what is the current situation? Let's take the 3 major geographies in which science is occurring and is increasingly occurring: USA, Europe, and China. And briefly, because we're past the hour, I'll just touch on some of these. And if there's time for a discussion afterwards, I'm hoping that we might be able to get other people's views on these subjects, because these are my personal views. Okay, in the USA, where I have probably more basis for my personal view, we've had a dramatic success following World War II, and we've had exponential growth that's driven that success. We've had a bottoms-up model. It's been individually initiated, not top-down. The exponential growth in our funding has allowed us to foster a bottoms-up rather than a top-down approach. As a result, American science across the board has been largely dominant around the world from the 1960s. But now things are changing. Our funding is steady or declining. In fact, the funding for biomedical research, by the most conservative estimate, has declined 30% in the last 10 years. In terms of the focus on the fundamental aspects of that research, in the long term, that decline is probably twofold again, that number. So there's an enormous negative impact. And what we have failed to do is transition to a new model, a steady state model that retains the key ingredients for success. In fact, we're going the opposite way towards top-down forced collaborations and short-term. And as a result, basic science is being dramatically reduced in my country, and funding is in all of the wrong directions. I, for one, hope that can be corrected, at least directionally, but I'm not especially optimistic. So the main question for the USA is, after 75 years after the transplant of Germanic science, who will the graft from Europe take ultimately, or will it be rejected? The US is known historically not for its theoretical culture, but for its pragmatic culture. We did not discover electricity and magnetism, nor the basic concepts of energy, but we developed the practical use of electricity, we developed hydrodynamic dams, and we developed the cotton gin. Okay? So we did not discover quantum mechanics, but we discovered semiconductors and the transistor. This is what America, left to itself, is really good at. We are a culture of engineers. Comes into America in the 1930s and 1940s, the great European tradition of fundamental science. That generation is now gone. The students of that generation are now gone. And the students of those generation of students are now my age. So we are the graft on the host of American pragmatism. Whether in the end American pragmatism will dominate and eradicate the German and European culture that was brought in, I don't know. But that is the big question in my mind for the success of American science long term. And I might say for the American economy. Europe. Well, I know less about this. I've learned more in the last, from discussing with a number of you the situation on this recent stay of several weeks. But it seems to me that Europe has recovered momentum after the losses in World War II steadily, but not explosively. If you look at the history of funding in Europe, and I mean principally the UK, France, and Germany, and different stories for different countries, but broadly speaking, a recovery after a major deficit, and then continuous but not explosive growth. And the result of that, perhaps with the exception of Germany and Switzerland, is chronic underfunding, for example, in this country. But there are certain advantages that you have. In the US, we would have regarded these as disadvantages, and they are disadvantages when it comes to exponential growth, namely the traditional university structures. The structures are stable, relatively top-down, relatively less fluid, with salaries that stem from the government or locally, and which to some extent exist independent of project-based funding. But it provides a level of stability. In good times, it's limiting, but in bad times, it's stabilizing. And so I think actually European science has an advantage right now for that reason. It has traditional disadvantages, but still advantages. I think, though, that the real question from my perspective for Europe, I'd be curious to know what some of you might think, is will the positive development of transnational funding schemes as Europe attempts to garner its forces, will European-based funding schemes, which are increasingly important, will they allow European science the benefits of scale, or will it in fact stifle innovation by forcing unnatural collaborations and fashionable top-down translational problems? Will it really actually, in the end, suck funds away from your stable national cultures to invest in short-term, what were ultimately less productive, economically also less productive approaches. And finally, China. Now, 5 years ago, China would not have deserved a mention on this slide. But with dimension comes mention. And China, as you know, is undergoing explosive increases in funding and research, truly explosive research. And they are emphasizing independent work by young scientists. So that is the emphasis. They don't always succeed in funneling the money to the young scientists, but it's their intent, and they are succeeding, from what I can tell, to a remarkable degree. However, they have a short-term reward system. So if you are a young Chinese scientist in a well-funded institute, better you should publish your paper in Cell, Nature, or Science. Better in many ways. First of all, your own personal bank account will be rewarded, the ultimate form of short-termism. You may earn the full equivalent of a year's salary for publishing a single paper in Cell, Nature, or Science. Now, that's a little less prevalent than it was, but it's really indicative. And what it's indicative of, I think, is short-term focus on a reward system that is driven by fashion. Okay, now what do I mean by fashion? I will relate to you a discussion I had with an institute director at a well-established but rapidly growing institute in a major city. And this director very proudly pointed out that his junior faculty had published 15 papers in Cell, Nature, and Science over the last 3 years, and they'd only been at his institute for 5. And how, by the way, was my department doing at Yale? Okay? And so the only thing I could say was, that sounds really exciting. I'm really happy that you're doing so well. Tell me about some of the topics that were covered by these papers. Well, every one of them had been recognized by a Nobel Prize in the previous 5 years. So it was guaranteed that these young people were not going to make a major discovery. They were focusing on the well-established and now very fashionable, okay? So what I said to the institute director was, this sounds like a good way forward for the moment, but if you keep this up and you have me back in 10 years, I wonder if you'll be able to tell me about the one discovery that actually mattered, that took place within your walls, because I can point to 2 Nobel Prizes that came out of my department. There were 2 major discoveries that were made. So it'll be very interesting to see how it works in China with this system. And doubly challenging is the fact that it's a patriarchal society that is lacking in transparency historically, and the current government, of course, is utterly lacking in transparency. They are trying in the science to put in barriers to the government culture that allow for transparency, but they haven't really fully succeeded. And I think, from my point of view, the big question in China is, for a culture that is historically patient, do they have the patience to build up science in the way that is necessary? You know, it's a culture of 5-year plans, and this is not a 5-year plan. It's not clear that any plan actually will produce the great cultures of European science, and now perhaps the great culture of American science. Were built up by historical accidents and by good taste and judgment, and by people over many years. They were not planned, as far as I know. So whether a planned society in 5-year increments is capable of, even if it does everything right, of achieving over a half a century of investment, a truly innovative culture remains to be seen. And so that is, I think, the major question for Chinese science, and the answer, of course, won't be known for many years, and probably most of us won't be around to observe it. But I, for one, do hope that China continues to invest and continues to develop in the direction that it has. So what I'd like to leave you with, that was somewhat pessimistic overall, because I do have a sense that we're in a time when, and I know many of you share this sense, that we're in a time when fundamental science is perhaps less appreciated and certainly less supported. We're all hopeful that this is a period in our development rather than what the long-term view holds. But as I look back on my life in science and the lives of scientists that I reviewed, other scientists that I reviewed for you this evening, I'm always impressed by the power of the scientific method. You know, those of us who have been fortunate to have contributed something that was notable in science, you know, you can say, oh, you know, I'm a very clever guy, and I have to admit, I used to think I was, okay? And I don't think I'm entirely stupid, but what I actually have come to the conclusion is that actually I'm not so smart, and actually none of us are. Somebody was very smart, probably somewhere between Galileo and Newton. They were very smart because they invented the scientific method. And the only thing it takes to really be a good scientist, I think, is to be in the right culture that supports and values the enterprise, to have an imagination, to be prepared to use it, and to stick to the scientific method with as little emotion as possible. And if you get too much emotion in there, you're not going to do as well as if you have less, and we all have some. So if the scientific method is constantly overwhelming me in its power, and it accounts for all of the success that I just related of the scientific enterprise, even in the narrow form that I just described to our benefit in public health. And for those of you who came to my first lecture, I had a quote from a scientific hero of mine, Buchner, who is the early biochemist at the turn of the 20th century, who showed that fermentation could take place in cell-free extracts, and who forever changed our view of life because we could think of life as chemistry. And what he wrote from his perspective, ever so long ago, is worth repeating. We must never let ourselves fall into thinking we shall never know, but we must have every confidence that the day will dawn when even those processes of life which are still a puzzle today will cease to be inaccessible to us natural scientists. Well, classic German double negatives, okay, maybe even triple negatives, okay? But the optimism that it expresses is extraordinary for a man of his time or any time. There is nothing that the scientific method cannot address. There is nothing that if we try hard enough, we won't know. Thank you very much. Before Alain asks for questions, I just want to point out, I've mentioned many collaborators and colleagues, probably about 300 who I've had the pleasure of working with over the last 30 years. And I've tried to encapsulate all of that into transport vesicles and fun, because I want to leave you with the idea that science is fun. And although it has its difficulties, we must always remember that science is fun, like blowing bubbles. So thank you.